Thanks everybody for your patience and sticking around and for tuning in today. Happy Friday as per usual. Today I'm really excited to introduce our seminar speaker, Dr. Lu Chou, who is currently a NASA postdoctoral program fellow at Goddard Space Flight Center and Georgetown University. Dr. Chou is an astrobiologist and organic geochemist whose research focuses on better understanding the preservation, detection, and characterization of organic biosignatures on Earth and other planetary bodies. She received her BS, or bachelor's degree in microbiology from the University of Maryland at College Park, and her PhD in Earth and Environmental Sciences from the University of Illinois at Chicago. And in 2019, she then started her current postdoc position, which is a joint position, as I mentioned, between Goddard and Georgetown, working with the Laboratory of Agnostic Biosignatures. Today we're going to be hearing about some of her work from her PhD looking at the preservation of biosignatures in briny environments in Antarctica. And so thanks again, Dr. Chou, for joining us and taking the time to chat. Take it away.
Great, awesome. Thank you so much, Dr. Riva Hernandez, for inviting me. And it's really exciting to be speaking with all of you today. For this particular audience, I'll mostly discuss the work that I did during my PhD, as said, at the University of Illinois at Chicago. But if there's a bit of time at the end, I can also give an overview of the work that I currently do as a postdoc at NASA on various flight projects and new efforts in astrobiology and life detection. And so the focus of this talk will be about biosignatures of life in cold subsurface brines, which are important astrobiology analogs for icy worlds like Mars, Europa, or Enceladus. Next slide, please. Okay. Now, cold saline brines provide a liquid oasis for microbial life that can thrive in really cold conditions. And it's these brines, they remain liquid even under cold temperatures due to their high salt content. And next slide, please. And it's this liquid oasis that can allow us to better understand how organisms can adapt and survive in such conditions, making it a really useful astrobiological analog for other encapsulated cryogenic brines that may be present under the polar caps of Mars or within the ice shelves of Europa. Now, several studies have already suggested that these liquid brines may be prevalent under the Martian surface and could be a source of the plumes putatively detected on Europa by the Galileo spacecraft. And the system that I will focus on today is Lake Vita brine, which is a fully frozen lake that encapsulates a cold hypersaline brine that is totally isolated from the atmosphere and has been so for millennia. Next slide, please. Now, Lake Vita is located in the McMurdo Dry Valleys in East Antarctica, as noted here in the red square on the top left part of the map. Next slide, please. And I highlight the location of Lake Vita, which is the northernmost basin in the Dry Valleys. The samples were collected in 2010, and all of the organic matter obtained in this time is the subject of this whole research. Now, I didn't actually go to Antarctica in 2010. I started grad school in 2013. It was all based on the research that my advisor, Dr. Fabian Koenig, had done at the time, and so he collected all of the samples that are presently studied. Next slide, please. So now I'll take a few minutes to highlight the objectives of this study. First, we wanted to study what biosignatures are produced, processed, and transformed in the Lake Vita brine ecosystem to better understand what kinds of biosignatures can be detected in cold hypersaline brines on other worlds. Next slide, please. And the second is what interpretations can we make regarding the presence of contemporary biosignatures, which are biomarkers of extant or modern life in Lake Vita brine, and whether or not these kinds of biosignatures are associated with modern life or potentially life in the past or ancient biosignatures. Next slide. And the third is, what are the current strategies that can be used to facilitate the detection and identification of organic biosignatures on other planets? Next slide. And to answer these three central questions, we use a number of interdisciplinary tools to interrogate the organic matter obtained from this cryogenic system. The first is metabolomics, which is the study of all metabolites in an ecosystem. You can think of this as kind of like think of the breakfast you had today. If I were to sample your stomach, I would be able to observe the biosignatures of the food that you, or rather your gut microbes, were able to break down and cycle. All of this material that's in a particular ecosystem at a time, at any given time, is termed metabolomics. Next slide, please. Metagenomics, on the other hand, is the total genetic information obtained from the environment. It allows us to observe the genetic potential of an ecosystem and thus the functional genes, as well as allow for the studies of microbial community. It's kind of like a book full of recipes for what you would make as an organism and how it functions, but instead of just looking at one organism, you look at the entire community. Next slide. Oh, yeah. And the third is geochemistry, which includes the physical factors such as temperature, pH, or salinity, and how they play a crucial role in how metabolomics, or metagenomics rather, can influence metabolomics. I integrated these 3 different platforms to investigate the biogeochemistry of the Lake Vita brine microbial assemblage. Next slide, please. The analytical methods that I used in this study includes conventional liquid-liquid extraction to obtain solvent-soluble metabolites, as well as lipid extractions from particulate organic matter. I then analyzed those metabolites using gas chromatography mass spectrometry. And quickly, for those who are unfamiliar, gas chromatography is an analytical tool that allows for the thermal separation of organic molecules. We can then detect them using mass spectrometry, which is an instrument that breaks molecules into smaller components, allowing us to look at the fragments and then putting them back together to get a whole picture of the molecule, kind of like putting together puzzle pieces to obtain their structural information. Next slide, please. Oh, thank you. I also use the multidimensional gas chromatography time of flight mass spectrometry, which is the more buffed up cousin of the GCMS. GCMS allows for the separation of complex mixtures of compounds based on their structural properties, whereas GCGC, or this multidimensional GC, allows for the secondary dimension separation of those molecules that cannot be separated using a typical GC alone. Here I show a chromatogram of a typical GC run, which may contain 3 peaks underneath that overlaps. With multidimensional GC, a modulator, a cold trap modulator, separates the mixture into a secondary dimension, and then those separated compounds are then sent into the mass spec. And these modulations can then be pieced back together to obtain the fully separated spectrum. And you'll be seeing a lot of these 3D plots today, but instead of having them as 3D, I'll actually show it as a 2D plot, or a bird's eye view, where each of the red spot is a peak and the blue is the background. Next slide, please. The metagenomic data was obtained by DNA extraction followed by next-generation sequencing. The data was then sent to the Joint Genome Institute for analysis and in-house gene annotation. And all of these DNA extraction was performed by our collaborator, Dr. Allison Murray, based at the Desert Research Institute. And lastly, for all geochemistry information, I obtained using previous studies of Lake Vita brine. Okay, so let's dig into the data. Next slide, please. Because we globally surveyed the solvent extractable metabolites of the brine, we opted to call these environmental metabolomics. Although we want to caveat this with the methods, with the fact that these methods target compounds that are more soluble in organic solvents like dichlorobenzene. There's a bias in what we're seeing. The liquid-liquid extract resulted in a metabolome extract that we then analyzed using a GC, showing this unresolved complex mixture, or UCM. And I show the retention time or separation time on the x-axis with the relative intensity on the y-axis. Now, unresolved complex mixtures are relatively common in GCMS studies, especially of older materials like crude oils and coals and things like that. And they're used to describe complicated samples that have many different constituents that overlap with each other, even in their retention time. But with some mass spectrometry, we are still able to identify some compounds based on ion chromatogram extractions. So these compounds that I found include chlorophyll derivatives. These are called malamides. Oops, I should say next up. Yeah, yep, malamides. Next slide, please. And the carotenoid derivatives are these norisoprenoids here. Next slide, please. And some of these are these loliolides or dihydroactinidiolydes. And I really apologize for the jargony terms, but the points will be made in a little bit. So while the unresolved complex mixture, next slide please, gave us some clues as to what some of the molecules in the metabolome of Lake Vita brine were, we were really fortunate to be able to separate the mixtures even further using multidimensional GC. Now this study only looks at a subset of these molecules because the list is exhaustive. As you can see, the number of peaks that are in the sample and their identification and confirmation of over 4,000 peaks is would have taken 20 years out of my piece, but I digress. Next slide, please. So upon multidimensional GC, we observed even more of these chlorophyll derivatives and carotenoid derivatives. Next slide, please. And the one after that. And so this was a bit perplexing. One before that. Thank you. This was a bit perplexing because we know that chlorophylls and carotenoids are photosynthetic pigments, which are not likely produced in the current aphotic, deeply isolated Lake Vita brine. Now, some non-photosynthetic microbes do produce pigments, but the diagenetic pathways that make some of these molecules seem to require atmospheric oxygen. So it was really confusing to observe their presence in this totally anoxic brine. So we then began to speculate that these could be remnants from the past. Some of these molecules, like these chlorophyll derivatives to the left, have been extensively studied, and their abundance in sediments is usually attributed to photooxidation or auto-oxidation from being exposed to the atmosphere. Next slide, please. And we opted to call these paleo metabolites. These paleo metabolites are representative of metabolites that were derived from a photosynthetic community. This compound to the right, the bacterial chlorophyll D, is a biomarker of Chlorobiaceae, which is a green sulfur bacteria that lives in the photic zone, the photic and euxinic zone of sedimentary basins. Next slide, please. And here are another example of metabolites deriving from pigments produced by diatoms like fucoxanthine and diodinoxanthines. And next slide. So why do we see these paleometabolites? Why do they exist in Lake Vita brine? We think that this is due to the slow metabolisms of the modern microbial community that takes a very long time to process the organic matter present in the environment. And so over time, these paleometabolites can accumulate and be mixed with the signals of modern metabolites. Next slide, please. Currently, the Lake Vita brine is aphotic, anoxic, extremely cold, and hypersaline. The microbiological survey of our system using 16S rRNA sequencing revealed that the system is exclusively dominated by bacteria, where no archaea or eukaryotes were detected. It was also determined based on radiolabel histidine incorporation experiments that the organisms in Vita have a generation time of the order of 120 years. So they grow super, super slowly. Next slide, please. But in the past, an ancient Lake Vita may have been more open to the atmosphere and supported by primary productivity, much like the other subglacial lakes currently present in the McMurdo Dry Valleys. Based on the presence of bacterial chlorophyll, one of the biomarkers that I mentioned, we think that this ancient lake may have had a photic zone euxinia and thus potentially a stratified water column or an anoxic sediment that had sunlight penetrated into it. So while the metabolomics of Lake Vita brine itself contains these paleometabolites that can confound our interpretation, we were still able to use it to extract information about past conditions that occupied the Lake Vita basin. Next slide, please. And so we describe these paleometabolites using the word legacy. This problem of legacy has already been recognized in Antarctica, where materials are often not recycled fast enough to allow us to distinguish modern signatures from past signatures. For example, radiocarbon usually takes a very long time to actually change its signature in Antarctica because of these legacy problems. And this is because everything moves at a very slow pace. Legacy is also, is it? Okay, sorry, I thought someone wanted to say something. I'll keep going. Legacy is also pervasive in the studies of lipids of the deep subsurface. An example is intact polar lipids that were routinely used as signatures of live biomass. However, it's known that potentially up to 96% of IPLs in the deep marine environments may be fossil molecules, and this legacy problem can possibly extend to other more labile organic matter, even like DNA or proteins. Next slide, please. So we conclude that these legacy metabolites may persist in other slow-growing systems, and thus the investigations of analog environments or any other important systems, such as the deep subsurface, should be considered carefully within the context of metabolic rates. For planetary exploration, especially if we're looking for extant biosignatures, things that are currently alive, we must consider the contribution of legacy when it comes to the evidence of extant life. Okay. That is the first major finding. Let's jump into the next one with the next slide, please. Great. So now, during the analysis of these paleometabolites, I stumble upon a series of polar organic molecules. The identifications of these molecules led to another publication because some of the compounds that were discovered have never been observed in other aquatic systems. Next slide, please. Okay, so here is the same multidimensional GC data set. We observed dimethylsulfone, a common volatile organic sulfur compound. And upon a closer look, we observed many more polar organic compounds, and we obtained the mass spectra from these compounds and was able to tentatively identify them based on extensive digging into the literature and comparison to the NIST library. Next slide, please. I'll walk through this method very quickly, and the data was obtained the same way as the previous study, but here, since we don't have the reported value in certain cases for mass spectral comparison using the database, we made the comparison ourselves by simply doing a linear correlation. This left here is the plot of the mass spectra observed for the compound suspected to be a sulfone, highlighted in the yellow in the chromatogram, and the bottom spectra is that of an authentic standard that doesn't exist in the library, but does exist in very old literature. We then calculated the percentage of each of the mass to the highest peaks and plotted the observed mass against the authentic standard, getting something that looks like a correlation. We then used least square regression to obtain an R-squared value. This example here shows the R-squared is 0.88, showing a positive correlation with a p-value of 10 to the minus 12, suggesting that we can tentatively assign this compound as a methane sulfonamide. Next slide, please. And all the way to the end of the slide. Okay, so using this method, we identified 19 more organic sulfones that have never been identified in any other natural ecosystems. These include the aliphatic sulfones, next slide, and yep, all the way to the end. Yeah. So these aliphatic sulfones, aminosulfones, cyclic sulfones, aromatic sulfones, and a few miscellaneous sulfones. They sound kind of like cell phone, my apologies. But among the organic sulfurs that we detected, there were two others that have these disulfide moiety. This compound here under miscellaneous, and one shown that's not yet to be identified. The study by Dr. Alison Murray also detected dimethyl sulfide and dimethyl sulfoxide, confirming the presence of some of these compounds that was detected in the same brine. But these were not actually observed in this study due to their high volatility and I guess unamenability to GCMS, especially if you're not targeting volatiles. Okay, next slide. Yep, okay, thank you. So with all of these detections, I was interested in the origin of these novel sulfones because they've not been seen in any other natural ecosystems. Now, we'll discuss some scenarios in which I think could explain the origin of these sulfones, although our understanding of these compounds are still pretty limited. But in order to do so, we first have to talk about how sulfur enters the biogeochemical cycle. So there are two pathways, the dissimilatory pathway and the assimilatory pathway. Most of you might be familiar with the dissimilatory pathway, in which sulfate is used in metabolism, where it is reduced to sulfide to obtain energy. Now, alternatively, sulfur can also be reduced to methionine and cysteine, the two sulfur-bearing amino acids, to obtain biomass in the assimilatory pathway. which is what we were interested in because we wanted to study the organic matter of the metabolites in the present in the system and whether or not those can constitute biosignatures. Next slide, please. Okay, this might take a minute. So here, I'm showing an extremely simplified schematic of the ancient Lake Vita. I do so by highlighting the pathways for the formation of dimethyl sulfone in lacustrine and other aquatic ecosystems. And then we can use this pathway to hypothesize about the formation of other organic sulfur compounds or other organic sulfones that was found in Lake Vita brine. So an inorganic sulfur, can be fixed into sulfur-bearing amino acids, methionine and cysteine, like I already mentioned, via phytoplankton biosynthesis. This then can be used to form dimethyl sulfonylpropionate, DMSP, a very common metabolite produced by algae in phytoplankton. DMSP is a precursor to dimethyl sulfide and other common atmospheric gases also present in aquatic environments. DMS can be oxidized then into DMSO and DMSO2 abiotically or biotically. And following the same pathway as DMSO, other sulfur-bearing organic compounds may also be oxidized to organic sulfones using this pathway. Now, the preservation of these DMSO and other organic sulfones into the current brine may be possible, and if this is the case, then the sulfones detected are also considered legacy. Next slide, please. Again, signatures of the past rather than the modern environments. Next slide, please. Now, alternatively, the dissolved organic matter that was retained from this past lake can also be sulfurized with reduced sulfur under anoxic conditions. There has been some evidence that suggests that water-rock reactions can promote oxidation of sulfur compounds, explaining the presence of sulfones in the current brine. This suggests that abiotic reactions can then play this huge role in the metabolite constituents of an environment at a given time. Next slide, please. And lastly, the third scenario, the sulfones themselves can be the metabolites of the modern Lake Vita brine microbial community. Although the scenario is particularly difficult to prove without conducting microcosm experiments, which is difficult for these really slow-growing organisms and would take another 120 years out of my graduate work. Next slide, please. Yeah. So based on our limited understanding of the formation of these sulfones, we concluded that these organic sulfones may constitute recalcitrant organic matter in aquatic environments, which have been previously to be predicted by many researchers who have also seen sulfone moiety in other marine DOM, or dissolved organic matter. Or they can be an abiotic alteration of product of water rock reactions or water brine reactions, or they can be metabolites themselves of the cold adapted bacteria that live in Lake Vita brine under in situ environmental conditions. So future research will hopefully be able to answer which of these three scenarios is most likely. Next slide, please. Okay, so I've discussed two whole research projects already where legacy dissolved organic matter is present and well mixed with the modern signature. Now, in order to easily distinguish those from modern signatures, we tried a different avenue where we looked at the particulate organic matter. So I'm showing here a zoomed in image of what the brine may look like. The particulate organic matter in this case is anything above 0.45 microns and less than 1,000 microns. And this would include most of the microbes that are currently living in the brine, which are the community that we are interested in. And from this process, we were able to obtain two fractions via sequential filtration, one on a 0.7 micron glass fiber filter and the other on a 0.22 micron PTFE filter. We then perform a lipidomic analysis, which is a subset of metabolomics that specifically focuses on lipids, and it is also more straightforward analytically. And we also perform scanning electron microscopy images of these filters to study the morphology of the particulate organic matter. Next slide, please. Yeah. Okay, so let's walk through some of the data. The lipids of the Lake Vita brine microbial community contains fatty acids. Not surprising. The dominant acids are the C16 and C18 in carbon chain lengths, which is very typical in bacteria. And as already mentioned, the microbial community in Lake Vita is exclusively bacteria. Remember that, exclusively bacteria. Next step, please. Yep. When taking a closer look at the others, we also observed branched fatty acids, monounsaturated fatty acids, and polyunsaturated fatty acids. which are not surprising given that these lipids or the unsaturations on these lipids are typically a result of the adaptive mechanism found in cold microbes. Next slide, please. Surprisingly, however, we found a series of sterols, which are lipid biomarkers or biomarkers of. Now, I included the structures of these lipids for those who are a big fan of organic geochemistry in the room. Next slide, please. And we also observed hopanoids and tetrahymenal, and their structures are shown here to the right. Next slide, please. Okay, now fatty acids are made by bacteria and eukaryotes. Sterols, on the other hand, are biomarkers of eukaryotes and can only be produced in the presence of oxygen. Another thing to remember, hopanoids can be produced anoxically by bacteria and some eukaryotes, but tetrahymenal is a biomarker of freshwater ciliate or a type of purple sulfur bacteria that lives in anoxic but photic zone of sedimentary basins. So the main question that I had was, who made what lipids and why are there sterols or these tetrahymenal in an anoxic ecosystem? The problem confused me for about 2 years, and so we decided to analyze these particulate organic matter directly via SEM. But first, I want to describe the brine microbial assemblage and what it's made out of. The filter I'm showing here is the PPFE filter, and you can see that it's loaded with these ultra small microbial cells. These sizes are extremely small compared to other aquatic systems and have been previously hypothesized by Dr. Emma Kuhn at the time, who was doing these studies of these microbial communities at DRI, to be cells that are reduced in size due to the environmental stress. And so, but just a quick note that these blobby-looking structure things are the PPFE filters, and the long, sharp, rod-shaped thingies here are not microbes. They're actually the glass fiber filters that we retain on a 0.7 micron filter. Okay, next slide, please. Many of these appear to also be dividing, so we really think that they are cells, but alternatively, they can also be vesicles that budded off of a cell. And the third potential interpretation is that they are viral particles that are being excreted from microbes. Next slide, please. Some of these are connected by some sort of filaments, and some are coated with extracellular polymeric substances, or EPS, which are, again, another indication that these microbial communities are adapted to cold conditions presently in Lake Vita brine. Next slide, please. When viewed on the glass fiber filter, which would have caught the larger cells, we also again observe these small microbial cells. They are essentially everywhere, and they dominate the microbial assemblage of the brine. But next slide, please. When we zoom out, however, we were just completely flabbergasted by these giant, large, cell-like looking things, these monster eukaryotic like detritus, and they look like they have seen better days for sure. Now, I want to mention that we speculate that these could potentially be old remnants of cells. We don't necessarily have the analytical tool to test whether they were, but they look like they are not living. Next slide, please. The size and structures of these are within the range of eukaryotes, and their shapes are very evocative of unicellular algae or protists. And next slide, please. And some of these detritus are rhombus in their shape and have these sharp apexes, which can help us identify them. But again, unfortunately, these are too degraded to definitively assign taxonomy based on their morphology alone. And again, they've clearly seen better days. So at this point, we have evidence in the dissolved organic matter and the particulate organic matter that Lake Vita brine is full of legacy, including these sterile lipids and these large, potentially eukaryotic cells that have died and degraded. But we think that some of the lipids in the lipidome itself can still be produced by the modern microbial community, especially those branched fatty acids and the unsaturated fatty acids that dominate the lipidome of the Lake Vita brine. So to address this question, we have to first determine if the brine bacteria can produce the legacy lipids, such as sterols. We know that if the detritus eukaryotes contribute the sterols to the lipidome, then the lipids are then considered legacy. But is it also possible for the current Vita brine to also produce sterols? There are always exceptions to the rule. And a good organic geochemist will tell you that sterols cannot be produced by bacteria, especially if they're in an anoxic environment. But again, microbes are very versatile, and there are always these exceptions to the rule. So I want to be absolutely sure whether or not the current brine can produce these sterols. And the answer might be in the metagenome. I first generated the list of the genes necessary to biosynthesize each group of molecules, the fatty acids, the sterols, and the hopanoids. And then I searched for these genes in the metagenome of the Lake Vita brine community. If the complete pathway is present, it means that the lipids could potentially be produced by the brine bacteria and thus be considered modern or extant biosignatures. If they are not, then they are unambiguously legacy. Next slide, please. Okay, so I show here the fatty acid biosynthesis pathway. The compounds shown are the substrates, intermediates, and products. And the arrows with the lettered symbols next to them are the enzymes that catalyze that particular reaction. Without discussing each of the steps, I simply highlight the presence or absence using a color code. So for any geochemist or microbiologist alike in the room or online, have no fear, you only need to pay attention to the colors of the arrows. Next slide, please. Okay, so here we detected all of the genes necessary to produce fatty acids from the Lake Vita brine metagenome. Again, this is not surprising given that bacteria are capable of producing fatty acids. I also detected the genes required to make branched and unsaturated fatty acids, but they are just not shown in this figure. Next slide, please. So we try to do the same thing for the genes required for sterol biosynthesis. I've removed some of the structures here for the sake of all of our sanity. And next slide. Again, while paying attention to the arrows, you can see that the complete pathway was not detected. There are many missing genes. And we only observe 2 genes in low abundance that can catalyze these 2 yellow reactions here, which is not enough to produce the sterols de novo. Yep. Hopanoid biosynthesis genes were also sparse. Next slide. Yep. With the squalene hopine cyclase gene, that particular gene was detected, but the genes to produce tetrahymenal or other hopanoids detected in the brine was not observed. Let's see, this gene here makes a 3-methylation, while in our sample we have a 2-methyl hopanoid. So it's a different gene that's necessary to make what we saw. Okay, next slide, please. So taken all together, we suggest that the sterols and hopanoids are legacy lipids. Sterols are likely produced by eukaryotic community, potentially photosynthetic, and hopanoids and tetrahymenal are legacy lipids, most likely produced by ancient bacteria or ciliates. Next slide. The fatty acids, on the other hand, can definitely be produced by the modern community, though we still can't rule out legacy. Some of those fatty acids themselves could also be a remnant of the past. And this was a really important point that we landed on because these fatty acids are the only evidence so far that we have of proper, I guess, extant biosignatures. And it was through metagenomic analysis that allowed us to confidently identify the potential modern biosignatures in these slow-growing ecosystems. Okay, next slide, please. All right, so now I want to come back to the idea of legacy, which is the whole entire point of this research. Legacy has been extensively identified in Antarctica, so the idea is not new or anything, but within the context of astrobiology and biogeochemistry, it is important to consider its implication. So I'm showing here a plot that was produced by Price and Sowers in 2004, which shows the average metabolic rate of communities of a variety of locations as a function of temperature. They obtained the data from a number of literature and generated this plot to show temperature dependence on metabolic conditions. Right, okay, so in a laboratory setting under in situ conditions, some communities from these environments, such as permafrost or polar oceans or ice or marine sediments, can grow relatively well, and their metabolic rate can fall along this the purple line, which is the growth metabolism line. Sometimes, however, even in the laboratory, in situ conditions can impose a biochemical constraint on the community. And instead of expending energy for growth metabolism, they allocate most of their energy on maintaining metabolism, which can include things like DNA or cell membrane repair. And this is present or can be present in glacial ice, Antarctic lake ice, or marine sediments. And lastly, the growth rates estimates based on geochemical techniques in situ to the environment itself show that some of these important studies can host organisms that are very close to their biochemical survival. Metabolism describes energy required to repair macromolecular damage. I mean, they are essentially doing everything they can to hold on just a little longer. And Price and Sauer speculated that this is much more representative of these important cryosphere environments and the deep subsurface itself. Next slide, please. Now, Lake Vita falls a little bit above maintenance metabolism, and this is because the microbes are living under hostile conditions that don't allow them for the opportunity for growth and development. And we were able to observe many of these leftover organic matter that was not degraded, both in the dissolved and in the particulate fraction. However, it does seem like the DNA was not necessarily subjected to this legacy issue, especially as I mentioned that I detected some of those fatty acid biosynthetic genes in the metagenome. Again, much like the accepted idea of preservation potential, DNA goes first, then lipids go last. Now, however, the slow metabolic rate that you get, the less and less organic matter is degraded and metabolism can stop completely. Aside from other abiotic factors, which can also be slow under cold limited conditions, molecules can persist over long timescales. And so as we move towards this survival line from growth, legacy likely to increase over time. And this is a really important caveat that should be addressed in all studies from those environments that are based on organic biosignatures, and particularly important for space exploration when we go out there to look for extant life. Next slide, please. Okay, so let's look at some of the major conclusions drawn from this study. The first one, next slide, please. The first one, what biosignatures are produced, processed, and transformed. We observed a variety of organic biosignatures, such as these paleo metabolites of surface-derived organisms, organic sulfones, potentially part of the recalcitrant organic matter, and these large detritus of legacy, potentially of possibly photosynthetic organisms. Functionalized lipids, as well as potentially modern lipids, such as those fatty acids. Next slide, please. Another objective that I had, and arguably the most important, is what interpretations can we make regarding the presence of contemporary biosignatures, and thus the occurrence of extant life in Lake Vita brine. We see that the legacy organic biosignatures are retained in both the dissolved and the particulate organic matter fraction. And again, this is all due to the slow metabolism of the extant microbial community and consequently slow turnover rate of the organic matter present. Next slide, please. And lastly, the third objective was to determine what current strategies can facilitate the detection and identification of organic biosignatures. We suggest that the investigators hold hypersaline systems, perform a system-level comparison with a multi-pronged approaches by looking at multiple different types of organic biosignatures, not just one, in order to confidently assess biogenicity. Now, luckily for planetary exploration, methods are already being developed for space-capable handheld or rover-held sequencing technology, such as the MinION or the search for extraterrestrial genomes. These kinds of technologies have already been established, and these could allow us to robustly investigate extant biosignatures. Next slide, please. So ultimately, for future direction, the results of this study point to the need to consider in situ growth rates while studying slow-growing, cold-limited, and isolated microbial communities. This metabolic flux, these arrows here, are very important to the standing crop of metabolites in an ecosystem. Next slide, please. And we are also interested in digging a little bit further into the metagenome. For example, we can't rule out that some of these fatty acids observed in the lipidome are now legacy, despite detecting all of the genes. And this is because metagenomic survey is a survey of all of the genes in a community, not necessarily all of the genes in one particular organism. But for the next step, we're interested in seeing whether or not these pathways exist in a particular organism to be sure which taxa can actually produce them de novo. And this can be done using metagenome assembled genome. And one last thing is, as far as space exploration is concerned, We know that cold-limited ecosystems are likely prevalent in the solar system. The result of this study informs the interpretations that we can make on return data that we'll get in the future. For example, when we get the opportunity to sample the subsurface brines of Mars, which will very much likely see the signatures of organisms that live in the previous ecosystem regime, potentially from the surface, that may be much different from what it is today. And our conclusion about the biogenicity of these biosignatures needs to be context dependent, especially using data obtained from genomic studies to confidently identify extant life. And that's it for the Lake Vita studies. If there is some time afterward, I'll be happy to talk about my current work. And next slide, please. I just wanted to give normal shout out and thanks to all of my collaborators, advisors, and students. Dr. Koenig raised me from a small, eager grad student astrobiologist wannabe to now a postdoc fellow working with Dr. Paul Mahaffey and Dr. Sarah Johnson at NASA Goddard and Georgetown, respectively, and other collaborators that have helped pave my scientific path here. So thank you so much. And of course, my friend NASA, NSF, the Earth and Space Science, and USRA. And happy to take any questions. Thank you.